Yeah. So today we'll start talking about termination, which is, I think, one of the most fascinating and interesting things that has come up in the last 10 years or so. And I wonder why it didn't come up earlier than that, because the fact that termination uh, of things like the neuronal signals and shutting off death itself, termination of life, it has been known for a long time. So and how it plays out in the immune system is something that is only about 10 years old. And it was a very interesting discovery and is a subject of a Nobel Prize. And that's why I wanted to talk about this topic separately. So what we've talked about and what we will talk about in the future are these things, right? So first, starting on the left, the first couple of lectures, we talked about recognition, the ability of the T cell receptor and the antibody molecule and the NK cells to recognize something that is foreign very specifically. That's sort of the first step in the immune response. The second is the activation. So that recognition, specific recognition, results in some kind of a activation signal, which then allows the cells to get activated. And then you have the effector cell response, where we looked at cytotoxicity and antibody production. So that activation leads to something. And just like we talked about in the analogy of society, when the army comes and destroys the terrorists in the city, right? Once the terrorists are destroyed, the city needs to go back to normal. That homeostasis needs to come back. How does it come back? Obviously, the immune response has to be terminated. All the soldiers that came into the city have to go back home, right? That's termination. That's termination of the immune response. And then whatever is left over is immune memory, which we will talk about when we talk about vaccines, etc. So that's sort of an overview of what the immune system, of the topics of the immune system we're going to talk about. So I'll tell you termination here in this slide that we've looked at earlier, and then tell you a little story of how it was discovered, how this process was discovered. So antigen taken up by antigen-presenting cells, presented on the surface of MHC molecules, delivers signal 1 through the T cell receptor and CD3 cytoplasmic tail. We talked about signal transduction. That results in activation of the T cells, which results in upregulation of certain molecules like CD40 ligand, which then deliver another signal through CD80-86 through CD28. And CD28 delivers signal 2. And then in shutting off the TGF-beta response on the T regulatory cell side, you finally get full signal of the T cell activation. So this is full activation of the T cell. Now that the response has been completed, and so terrorists have been wiped out, so to speak, in the analogy, things need to shut down. So what happens immunologically is another molecule is upregulated much later in the kinetics of the immune response. It's called CTLA4. And CTLA4 has a 20-fold higher affinity to CD80 and 86 than CD28. So when CTLA4 is expressed, then it preferentially binds to CD80-86 over CD28, even though both molecules are present at the same time on the cell surface. And because it preferentially acts on the cell surface, it delivers a new signal, and that signal is the off signal, which is the surprising thing. And where I'll tell you what the surprising thing is like this. Just before the class, I tried to make a figure, and this is all I could come up with. But to tell you the story of how this was happening, you have to go back to, let's say, 1990s. So 1990 or so. So if you remember the sort of little bit of immunology of history of immunology, DNA was discovered in 1950s, 1958 or so, Watson and Crick. So that was 1950s, mid 1950s, and then in 1960s, people started understanding that there may be something like an immune system, and then by 1970, another 20 years later, the fact that we make antibodies and that maybe T cells and B cells were being discovered, And in the 1980s, more molecular understanding of that antibody molecule, the rearrangement, immunoglobulin rearrangement, discovery of the T cell receptor, all that was happening. CD4, CD8 molecules, all of these molecules are being discovered in the 1980s. And then how T cells are getting activated is being discovered. All the functions of these CD molecules are being discovered. So a very simple experiment was done. Well, this is the kind of typical experiments that were done in vitro experiments were done in the 1980s and 90s. So you would take lymphocytes from a patient or a healthy individual and stimulate the cells with anti-CD3 antibody, which essentially stimulates the T cell receptor, right? And on the y-axis would be either interferon gamma secretion, it could be cell proliferation, any positive signal that you can think of, right? And you would get certain level of, let's say interferon gamma. Let's say there'll be certain amount of interferon gamma that is secreted. Then what you would do is because you know that co-stimulation is involved, you would stimulate the cell with a combination of CD3 and anti-CD28. It's a combination. We need two signals, right? So give both the signals at once. And then you would double the signal. So that signal of 10 would become signal of 20. And then when CTLA4 was discovered, everybody said, okay, let's do the same thing. Let's add anti-CTLA4 antibody into the culture with CD3, combination of CD3, CD28, and CTLA4. And lo and behold, you would get a huge immune response when you added anti-CTLA4, right? So if I go back to the previous slide, and I stop here, you have your signal 1 and signal 2, right? Which delivers a very strong signal, and CTLA4 is expressed, and CTLA4 delivers that signal 3. If in vitro experiments were the only experiments that were done, the resulting response is a massive immune response in vitro. So everybody said, signal 3 is very important because signal 3 is a positive signal and induces this huge amount of cytokine production by the T cell. That's what you would do with in vitro experiment, right? But what Jim Allison did, and we will talk much more in detail about this when we talk about immuno-oncology later. There'll be a lecture, we will talk only about immuno-oncology, and that time I'll show this thing again. But today, what I'm going to show you is that Jim Allison in 1996 published a very nice paper where he and his colleagues in his lab showed that if you take mice and you induce a tumor in these mice, in the control mice, which are shown in the hollow circles here in the middle, and in time, that tumor just keeps growing if you don't do anything to the mouse, right? This is what you would expect. Now, dogma at that time was if you stimulate them with anti-CD28, it should deliver that signal 2, very strong signal 2, and the two signals should activate the T cell, and those T cells should be able to kill the tumor because you've activated the T cells, right? But interestingly, if you give anti-CD28 to the mice, not only do the animals not eliminate their tumor, but they actually augment that tumor. The tumor even becomes worse, it becomes faster, right? And CTLA4, when you give anti-CTLA4, see what happens. The tumor vanishes. It's a very unusual observation. Everybody said, CTLA4 induces a positive signal, and that's why you're getting huge activation of T cells, and that's what's probably happening. Until such time, it was clearly demonstrated that CTLA4 doesn't have a negative signal, but induces a positive signal. So what is this? How does a negative signal, which is shutting off the immune response, responsible for eliciting such a strong T cell? It's almost, saying the opposite thing. So in order to show that, let me open that clinical immunology or the immuno-oncology slide, because I have more detail in that. I'll show you there. What the experiment that was done was like this. So this is the same slide that I showed you earlier. And this is the X-ray graph of a mammogram where you can see a large tumor on the left and treated with this patient treated with anti-CTLA4 antibody, the tumor completely vanishes. So what was seen in mice, is dramatically seen in humans also. So what was happening? So if you look at the same picture again, antigen taken up by antigen-presenting cells, deliver signal 1, signal 2, TGF-beta, you get your full signal in your T cell activation. And this is your tumor, by the way. On the right side is the tumor. And so if you get normal activation of the T cell, and this is what happens all the time in our body. When people, we get mutations all the time in our body that are happening, and our immune system constantly is doing surveillance, looking for mutations. And if there is a mutated cell, it recognizes that mutant cell and kills it. This is happening. The elimination phase of the body is happening all the time, right? Now, in addition to CTLA4, there are other molecules called PD-1. PD-1 is just like CTLA4, it's just a different molecule. Now, interestingly, that PD-1 also does the same thing like CTLA4, which shuts off the immune system. But now the tumor has got smarter. Now the tumor, the remaining tumor that was not eliminated by the T cell, starts expressing the ligand for PD-1. PD-1's ligand is called PD-L1. And PD stands for
program death.
Program death ligand 1. Yes, program death. So again, look at now the difference between the previous slide and this slide is that the tumor endogenous now expresses PD-L1 just like that, right? So antigen taken up by antigen presenting cells, signal 1, signal 2, full T cell activation. Now what happens is PD-1 is expressed on the cell surface. And because PD-L1 is being expressed by the tumor PD-L1 binds to PD-1 and delivers the negative signal to T cell. And so the T cell and the immune response shuts off prematurely. And so the tumor cannot be killed by the T cell. And that's why you get cancer. That's why cancer progresses. Because the tumor is sort of, I would say, not intelligent, but by expressing PD-L1, delivers the negative signal into T cells and shuts the T cell off. So the therapy for that is when you give a therapy which blocks the PD-L1 from binding to PD-2. This is a monoclonal antibody, right? You treat the patient with a monoclonal antibody that blocks this interaction. You go through the same signal 1, signal 2, signal 3 process. Now, when you deliver signal 3, that antibody has blocked the negative signal, and the tumor can be eliminated now. This drug is called Keytruda, made by this anti-PD-1 antibody. The drug is called Keytruda. The generic name is pembrolizumab, and this drug makes close to $20-25 billion a year for markets, transforming how tumors are being treated. Almost 30% of the patients who get this drug are completely cured of their tumor. You treat patients with anti-PD-1 antibody, and the immune system is able to kill the tumor. So this is a revolution in the field of immunology. And this will go back to the termination slide deck. The discovery was made by Jim Allison to show that it was the negative signal. So what was happening here in his experiment is that the T cells, the tumor was not allowing the T cells to kill by expressing the inhibitory signal. But then when you block that inhibitory signal, by CTLA4, they were able to get the elimination of the tumor. And for this discovery, Jim Allison, who discovered CTLA4 interaction, got the Nobel Prize, and PD-1, PD-L1 was got by Tatsu Honjo in Japan, and they both shared the Nobel Prize for this discovery. So let me pause here and see if you understood that, because this is the main topic for immune termination. We can talk more about it. Any questions or comments, or this is what I wanted to share.
So, Naren, I have a question. So in this case, like
when you started with like the word termination, I thought how the body actually stops activating the T cell, like signaling pathways and everything, right? But this termination is different from like the termination, or because this is very different, right? In this case, the body is actually the tumor cells are growing, and then with the help of PD1 and CTLA4, they are actually like gaining immunity versus the immune cells. So like these are two different types of termination, right?
Yeah. So in the normal situation, when you don't have a cancer or you don't have viral infection, or the virus of the tumor is not interfering with the mechanisms of your immune system, when you get infected with the influenza virus, for example, there will be a huge expansion of your T cells. These T cells will invade the lung. They will kill the virally infected cells. And once the virally infected cells are there, the expanded immune cells that are there have to go away.
Yes,
That is
that makes
normal
sense.
termination that happens all the time in our body. And that is through apoptosis. A majority of those cells will die off. Like we talked about in apoptosis, they will activate the caspase and they will just dissolve and vanish.
Okay. So in
That
this
happens
case,
normally.
basically, yeah. Okay.
But what happens
Now I
in tumor is tumor hijacks the termination signaling process
yes.
from the immune system and shuts off the immune system's ability to kill the tumor. And that's how the tumor survives.
Okay, so these are the two main receptors which are involved in like hijacking the immune system,
Yes,
right?
these are the two main receptors for which there are drugs available. There are literally hundreds of more antibodies like this. And as Praveen mentioned in the chat, these class of antibodies or drugs that block inhibitory signals are called checkpoint inhibitors.
Okay, I see. Thank you.
Yeah, good. All right.
I wanted to ask, why are they particularly called checkpoint inhibitors? And
I don't really know where that term came from, but I think it is in the pathway of the signaling cascade. It may be a point where the I mean, I don't know. I'm hand waving. I have no idea. Maybe we can Google it and find out why they are called checkpoint inhibitors.
yeah.
Maybe like from chess, like checkmate.
So they
Checkmate.
Yeah.
Checkmate.
It's like,
I don't know. I don't know about that. Yeah, that's something we could look up. Yeah.
they are the protein which are involved in the immune system of the cells. So we need to block that protein so that there is no transfer of signal.
Yeah, so the signals are blocked. So to follow up on what Pushpam just said, CTLA4 binding to CD86, PD1, PDL1 interactions are the two drugs, the two interactions that are involved. And there are many more interactions for which drugs are in development right now. Clinical trials are ongoing. For example, TIM3, TIGIT, LAG3, all of these are, the names you don't have to remember all the names, but these are ligand and receptor pairs. Again, they all deliver negative signals into T cells. They all fall into that class of checkpoint inhibitors. And one of the key points that, because it's mentioned on this slide, is they have a signaling domain which activates the phosphatase. And I think I have a slide on that, but yeah, this is another slide showing the same thing, all the negative simulators of T cells. I think I've talked to you about this earlier. I don't know whether I've showed this slide to you. I like to draw. So I take a big chart paper and then draw all the signaling cascade. I showed you one for the CAR T cell earlier. This is the same thing. So that's one mechanism of termination of the immune response, or the immune response shuts down during the role of the immune response. This next phenomenon, which is an important phenomenon that occurs in T cell activation, is a term called exhaustion. And this just like us, if we run too much, we'll be exhausted. If T cells get activated for too long, they will get exhausted. And the exhausted phenomenon is basically at a high level, exhaustion of the metabolic capabilities of those cells. So all their metabolic capabilities are lost, and therefore they become non-functional. They might be there, but they're not dead, but they're non-functional. And that's a phenomenon that is called exhaustion. And that happens a lot in cancer again, because in cancer, the cancer is, omnipresent, and there are lots of tumor cells and T cells, and the immune system is constantly trying to get activated and kill these tumor cells. And then, at some point, these T cells get active. These T cells get exhausted. A huge, if you're interested, we can talk much more detail about exhausted T cells in one of the upcoming classes later. But it is a very deep subject which requires much more, reading. But I just want to introduce the topic to you. One of the people who's done the most amount of work in this area is a professor, John Very. He's here at University of Pennsylvania, and he's classified these exhausted cells into various stages of exhaustion. So exhaustion also occurs in a staged manner, and these are all the transcription factors that are involved in the transition to exhausted cells, right? These are more about exhausted cells. So that's sort of the termination of the immune response. Again, like I said earlier, we will discuss all of these points from innate to adaptive to cytokines to signal transduction to termination of the immune response when we start talking about case studies, which we will start next week. Okay. The final topic in this immune regulation is tissues. So for tissues, I have to tell you a little story to understand why tissues are important. And this realization that the immune response in different tissues occurs very differently is, again, not a very old phenomenon. People knew about it, but really focused on developing the processes to understand it, only in the last decade or so. So to give the analogy of what happens in the tissues to frostbite, that's shown here on the left. So some of you must have got a frostbite at some time in your life. You touch a dry ice or very cold piece of material, and then you can see on the left your fingers turn white, right? And it's not like burn. I mean, you can touch a very, very cold thing for at least a few seconds, maybe even a minute sometimes, right? A hot thing you can't touch even for a second. If it's hot, you immediately remove your hand. But a cold thing, you might put your hand a little bit longer, especially like if you're in a cold region or in the snow, walking in the snow for too long, you can get a frostbite. So what is the analogy of frostbite and the immune response? And this is it. So what happens during the frostbite is that capillaries dilate to keep the extremities warm. So when you put your hand in a dry ice, and you put these two fingers into dry ice, the first thing that will happen is your capillaries in that region will go towards that and provide more blood to that region, so that the warmth of the body will reach the region that is being burned, that is getting that frostbite, and try to keep it warm, right? But if you keep the finger there for too long, right, then what happens? Then the capillaries start restricting. They will start constricting. They will start moving away from there. Initially, there'll be dilation, but later on there'll be constriction, because they know that this is not going to help. And if the capillaries keep dilating and allowing the blood to flow there to keep the finger warm, and if you're and that warmth is not being solved, but the cold is still coming, that cold that the body is experiencing could traverse through your entire body, and your entire body will go into hypothermia from that source of cold. So what the body does is, instead of allowing the body to die, the body will say, to hell with it, let me constrict my these blood vessels. I'll lose my fingers because of I'm constricting my blood, and all the nerves and the blood vessels are all constricting, and I'll lose my fingers because of the frostbite. but at least I won't die. Right? I won't die of hypothermia because of that. So that's what happens. That's why you lose your fingers during frostbite, extreme frostbite. So what happens in the tissue? So what happens in the tissue for an immune system is when you get infected by the liver, for example, when you get hepatitis infection in the liver, and we know that the liver regenerates, isn't it? Liver is a regenerating organ. So when you get infected with hepatitis B in the liver, the immune system will activate cytotoxic T cells, CD8 cytotoxic T cells. These cytotoxic T cells will invade the liver, and through cytotoxic killing of the hepatocytes, because the virus is inside the hepatocytes, they will kill off all the hepatocytes. You will get severe jaundice because of some infection, because your liver is being killed, right? But it's okay, because once the liver is killed and the virus is eliminated, because of the regenerative capacity of the liver will be back to normal after the virus is eliminated. So cytotoxic T cell in the liver is a very common thing. Okay. Now, if you get infected in the eye with the cytomegalovirus, another virus which commonly infects the eye, the body doesn't activate cytotoxic T cells. Same kind of virus, similar kind of virus, same kind of immune system. You can activate cytotoxic T cells and eliminate the virus in the eye, but the body doesn't do that because the eye doesn't regenerate. And because the eye doesn't regenerate, the immune response that is elicited in the eye is very different from what happens in the liver. You actually activate what is called a Th17 kind of response, which is a different kind of immune response. Different set of cytokines are involved. And by secreting different kinds of non-cytolytic enzyme, meaning immune responses which don't have cytotoxic activity, they have immunosuppressive activity. So they are able to suppress the immune response in such a way that that the virus replication is suppressed, but not killed. The cell don't die. One second. Yeah. So in the eye, antiviral infections are not cytotoxic, and this is very important. However, if the cytomegalovirus decides and is not being eliminated by the immune system, and the and that virus remains in the eye, then later on a cytotoxic T cell response will be induced, and the eye will be destroyed. Similar to the frostbite story, if you remember, right? First, they'll try to remove the virus without cytotoxicity. And so what is the kind of the immune response that happens in the eye? You get activation of TGF-beta, VIP, anti-MSH, TSLP, RA, all of these immunosuppressive responses. T-regulatory cells are involved. Typically when you've probably heard about this is IgG in the liver or in the muscle or in the kidney or all of these other organs, you will get IgG production. But in the eye, you get IgA. The nature of the immune response is very different in the eye. And Bhavya, I think this is why I want to write the immunology of the eye, because I think the immunology of the eye is very different from the immunology in the liver. Right? So that's something that one of the reasons we are doing this course is to be able to have a better understanding of immunology so that we can start writing about the immunology of the eye. So that's sort of the eye story. And so this finishes our immune regulation overview, where we covered innate immunity, adaptive immunity, cytokines, signal transduction, cytotoxicity, termination, and the nature of the tissue-specific immune response being different. The kind of response that is elicited is dependent on the tumor, is dependent on the tissue. And as you can imagine, because if it is tissue, tumor is also a tissue. So a liver cancer immune response is very different from a kidney tumor, is very different from a breast cancer. So all tumors have very specific immune responses, and the nature of the immune responses is determined by the nature of immune cells in that particular tumor. Okay. All right. So I think we've finally finished the immune regulation lecture. It took 4 or 5 sessions for us, but I think the short bursts have been good, at least for me.
Right. I wanted to ask something that you mentioned yesterday about complement activation,
Yes.
whether it's an effector function. So what is actually an effector function? What is it?
Yeah. So I don't have, I might have slides somewhere, but maybe we'll do that in a future session. It might be interesting for you to listen to the, or we might discuss the complement activation when we talk about patients who don't have complement. If they don't have complement, certain complement protein, genetically, if they are missing some components of the complement protein, they get some specific kind of bacterial infection. So that will allow you to understand how complement works. But essentially what is complement? Complement is a series of enzymes, much like the enzymes that are involved in clotting of the blood. So when blood clotting happens, you have all these factors, clotting factors, which are called factor 1, 2, 3, 4, 5, 6, 7, 8, right? And all of these factors are not active in our blood until we get some kind of a cut. When you get a cut, then fibrin and fibrinogen, which are 2 proteins that are involved in sort of forming the mesh over there, activate factor 1. I don't know the sequence, but let's say they activate factor 1. When factor 1 is activated, then factor 2 will be activated. Factor 2 is activated. Factor 2 activates 3, activates 4, activates 5. Finally, you get all the factors activated, and then they form the clot. And that's how you get clotting. Similarly, complement is also made up of complement proteins. So you have C1, C2, C3, C4, C5, C10, etc. You have this whole complement cascade, just like the clotting cascade. And for simplicity, I'm just going to tell you, when a bacteria comes into the body, it activates C1 and activates complement 1. When complement 1 is activated, then it will activate complement 2, 3, 4, 5, 6, 7, 8. And then what happens is that complement numbers 5, 6, 7, 8, 5, 6, 7, 8, 9, the last 4 of them, they actually, like, if you remember perforin protein, which made pores into the body, into the bacterial membrane for cytotoxicity. Complement 5, 6, 7, 8 are like perforin. They make a pore and create an osmotic imbalance in the bacteria, and they kill the bacteria. So this enzymatic cascade of complement proteins, is a very primitive immune system. This is before when T and B cells came into being. It's very old, but we also have it. Like humans also have complement systems. That's complement.
Okay. It's called complement system because it's enzymes and not like, but they are also signals, right? So it's just.
They're not signals. They're just one enzyme activates the next enzyme. There's no cell involved. They're extracellular. They're free flowing in our serum.
Got it.
Yeah, I'll show you a slide about them. The reason I don't want to tell you, I don't want to quickly show you something right now because it'll confuse the matter, because the complement cascade, while sounds really nice to tell you that C1 activates C2, activates C3, activates C4, etc., it's not that linear, because just for historical and nomenclature purposes, actually you first get activation of C1, then which activates C3, then which activates C2, then you get 4, and then you get 5, 6, 7, 8 together, and it's like a cadence. Like it's not nice, neat, and clean. it's a little convoluted, but the idea is that you get one activation, you get activation of what is called the effector activation of the cascade, and complements 5, 6, 7, 8 form the pore. The pore literally bores a hole into the bacterial wall, and then osmotic imbalance happens and the bacteria rupture.
Okay, so when, I mean, even in the video, right, you said when apoptosis happens at regular times, I mean, all the I mean, it bubbled out and it's just recycled inside the body. But when it's a foreign cell, what happens to the waste? I
Well,
mean,
good question, good question. The waste obviously has to be cleared. So what happens then is the macrophages, the monocytes, maybe also in the brain, they're the glial cells. In the liver, they're called Kupffer cells. These are all macrophage lineage cells. They're like amoeba. They are like engulf all of this stuff and eat it up. They digest all of this inflammatory material.
okay. All right. And
They're
last,
scavengers, like these are scavengers, like the garbage collectors, so to speak.
and lastly, in tissues, I mean, it says there are 3 phases. So first is doing nothing. So is it like innate? I mean, it's not doing nothing, but it's just well, what's the phase 1 in these 3
Yeah,
phases?
phase one in need to be like, there's a little bit of sensing and making sure that the organism or the cell that you're going to try to attack is really the right person to kill. Like, I want to make sure that I'm not killing my own body cells. I want to make sure that whatever I kill, whatever I'm getting ready to kill, better be a foreign thing, which is not part of self, right? If it is self, I don't want to kill. Otherwise, I'm going to get autoimmunity.
Right.
When things go wrong, then I'm going to start killing my own cells of my own body. And we'll study autoimmunity as one of the chapters in the course. But that's, the innate part is just making sure that I'm going to kill the right thing. I don't want to kill the wrong thing.
Yeah. Got it. So that's phase 1. And then 2 is trying to solve the problem with as little damage as possible.
As little damage as possible, and it is highly regulated that I'm going to give you one signal. I'm going to give you one punch, and maybe it'll knock you out a little bit. But then if you're really foreign, I'm going to give you that second punch, and then maybe I'll give you a third punch, and then you will die.
Right.
I'm not going to kill you like I'm not going to throw a rocket at you the first time I see you.
This is only with tissues, right? When it's something else, it's blood or something else, and there's a
There also, there also, for example, if an HIV infected cell is circulating in the body, then the immune system will hunt it down and kill it. Just
right.
like it would be in the tissue.
But you're teaching tissue separately because it's somehow different, right? It's not
somehow, it's somehow different because.
So what is the difference?
So where the field is figuring it out is that it is not just the immune cells that are responsible for eliciting the strength of the immune response. Like I've been telling you, antigen presenting cells like macrophages will induce activation by stimulating T cells with signal 1, signal 2, signal transduction, cytokine secretion. It sounds really nice and neat, and I can tie a bow on it and say, okay, now you've learned immunology, right? In reality, what happens, there are hundreds of different other cell types that are not macrophages and T cells. There's like these endothelial cells, there are fibroblasts, there are mesenchymal cells, there are platelets, these cells also doing all kinds of crap.
Right.
And influencing the immune system, right? To give another analogy, maybe I don't know if it's the right analogy for this group, but because the analogy is going to be for the brain, like, we have hundreds of hormones, right? We have epinephrine, norepinephrine, we have oxytocin, we have dopamine, we have this and that and the other, right? I can tell you, I'll give you dopamine and you'll feel really happy or whatever, right? It's happy you because of some part of the brain that is activated. But it's never like simple, right? I can't just give you dopamine and everything. Some oxytocin will come and some epinephrine will come and some adrenaline will and mess up your dopamine dose, right? So if I give you dopamine, you may feel happy. If I give dopamine to somebody else, they might feel something completely different because of the nature of the other hormones that they have. Similarly, every organ has a different repertoire and a combination of cells and cytokines that are secreted, and the overall nature of that environment that is created results in the final elicitation of the immune response that happens in that organ, right? And that's individual to individual different, organ to organ different, organ to organ different. Every virus induces a different kind of immune response. Cytomegalovirus, because the nature of the antigens are different, the genes are different, so many. And plus, in all of this, we will talk about this, what the hell the gut microbiota does, we have no idea. So I'll just introduce the gut microbiota story now, because I told you that Keytruda story, right? When I told you about Keytruda, or the anti-PD-1 or the CTLA-4 antibody, I told you when you treat animals with anti-CTLA-4 antibody, or you treat humans with anti-PD-1 antibody, like I showed you, blocking of that termination signal activates the immune system, which then kills the tumor, and it's, and everyone lives happily ever after, sort of, right? The great story to tell, right? There are now a bunch of papers that are coming out in the last literally couple of 2-3 years, that if you do the same experiment, and the only thing that you do is you remove the gut microbiota of the mouse, in that same experiment that I showed you for Jim Allison experiment where the tumor goes away, if all you do is remove the gut microbiota, anti-CTLA-4 doesn't kill the tumor. So what the gut microbiota is doing to something that is completely unrelated to obvious things, we don't know yet, but obviously is doing something very important. But we don't know what. So the study of gut microbiota and the role and in the regulation of the immune response is a very big field that is emerging.
When you say remove it, I mean, how do you remove?
Well, in a mouse, you can give antibiotic. In a mouse, you can give antibiotic. In humans, it's more difficult. But in mouse, you give antibiotic and you sort of clear the gut microbiota over there with this antibiotic. Yeah. So anyway, still, even though we are starting to learn, like I told you initially, we know immunology, but there's much more that we don't know than what we know. But let's figure out what we know first, and then we'll uncover things that we don't know later. All right. Any other questions? Any other questions from anyone else? Abhavya, you can also ask. This is great.
I wanted to ask, as we discussed yesterday, that everybody has different kind of cytokine responses and all. And we now know that tissues also have different kind of responses.
Correct.
So, will we study specific tissues and as well as the pathologies related to them? And
I think it's very important. I think it's very important to study immune response in the context of the tissue that you're studying. Like you're working on CAR T cells, for example, right now, right? In immunity, right? And you know that that CAR T cells works really well in in the people it works in. It works really well in blood cancer, like leukemias and stuff like that, right?
Yeah.
CAR T cells don't work in prostate cancer and ovarian cancer, and it doesn't work.
Yeah.
And there are many reasons it doesn't work. One is that the CAR T cells may not be entering those organs very well, but there are also studies where they show the CAR T cell actually enters the solid tumor, but is still unable to kill. Because the nature of the tissue is very different. We have to study what's happening in the tissue. The tumor microenvironment is playing a big role in the kind of the immune response that the CAR T cell generates. So lots needs to be studied.
Yeah, so they are finding it difficult to break through solid tumors.
Yes.
But I guess there is a lot of background fundamental research in that sense that still has to get done.
Get down, exactly. And we are just discovering what we are going to do in our class, hopefully. And when we write when we start writing the paper on immunology, because we don't have a lab, we are not working on trying to solve the problem. I think if you can even in the paper, if you can even define what the problem is, that is a good paper. You don't have to write the answer.
Right.
Find the question also is good enough. Yeah, yeah. For me, I've been studying immunology. It's like amazingly fascinating for me. Hopefully, the fact that you guys are calling on week and not missing any class, I feel like you're also getting it slowly.
I'm more confused than I'm clear about it. Yeah.
No, that's okay. That's okay. That's okay. Absolutely okay. What I will say is, I don't know if we want to do this or not, but I just thought of it. Do you think it'll be a good idea for you, all of you, maybe in the next week or next 2 weeks or so, instead of me presenting, you make 5 slides and just tell a story that you've understood well. Maybe each one of you just take anything. It doesn't have to be the whole thing. You can say, okay, this was fun. Let me try to, and you can take my slides only, or you can take something from Google or whatever, and just tell one story, 5-6 slides. Does that sound good?
Yeah.
Like a little
So one
exercise.
story regarding like any case studies on the disease, or
No, anything you want. Anything you want. And like what I like is it doesn't even have to be a conclusive story that this is a beautiful story. Like I started here, I didn't know this is what I found out, and this is what the solution was, and this is how everything happened. You can present something that I'm presenting something, I don't know what the hell I'm talking about, and at the end of the presentation you can say, this is all that I didn't understand. That's also a good presentation. Because we'll all listen to each other and say, okay, all of you may have answers to that. Just take anything, and don't look for perfection. Make it as confusing as it is actually in your head.
Sounds good.
Present the confusion. All right, so we'll do that next week. That sounds fun to me, though. So to listen to what you guys might come up with, and next Friday we'll try to do that and see what happens.
so all of us are doing it next Friday, right?
Yeah, if you can.
Yeah, sure.
There's no obligation.
All of us
No
in the
pressure.
same.
Hmm?
All of us are on the same day or different?
We'll just do one by one and see if we do it, we do it. If it goes smoothly, we do it. I think what might happen is, Bhavya will present her 5 slides. She'll have 20,000 questions, and the whole time we'll be talking about her questions only. Then we can do the next one next week. But prepare for something and see what happens. This is just
I think it would be like. I think it would be like some live exam we are giving.
Okay. Consider it an exam. What the heck?
Yeah.
Like, but then if that's putting pressure on you, then don't.
Yeah, exactly. If it's putting pressure on you, don't do that. Do it like it's an exam. Do it with a little bit of pressure, but don't go nuts.
Yeah.
Important to mention that this whole thing has to be low pressure. I mean,
Yeah, and also use Google, use textbooks, talk to your friends and see what they are saying, and come up with a story that it also will allow me and also you to know what you've actually understood so far.